shorter duration of disease, less opioids, et cetera, correlated with ultimately better outcomes from a pain perspective. And so the question is how do we define when is the right time? Because the longer a patient is on opioids, some proportion of those patients are going to end up with central sensitization. And once you reach central sensitization, the horse is out of the proverbial barn. Thanks.
Excellent. Thank you, Jamie. We would move to our next online speaker, Dr. Sohail Husain, a great colleague and friend and a mentor even to me in the field. Sohail is from Stanford University School of Medicine. We'll be talking about tackling the global problem of drug adverse events using the prototypic example of asparaginase-associated pancreatitis.
Great, thank you. Firstly, can you all hear me?
Yes, we do.
Okay, great. And secondly, this is going to be an experiment. If this works well, give me 30 seconds to get this started. If this works well, we'll run with this. Otherwise, I've got a recording in case there's a problem. So give me 10 more seconds here. All right, can you see this?
Yes.
Yes.
Okay, perfect. All right, well, thank you, Maisam, for the introduction. Peter, Miklos, Andrea, and all the organizers for the invitation, for your courage to hold the 10th conference of the Hungarian Pancreatic Study Group. This acknowledgement slide at the outset attests to the value and breadth of team science. It highlights many people, including Maisam, who's here with us today. I'd also recognize Olivia Sai, who leads the project I'll tell you about, and Toshi Sato, who stewarded much of the omics data. The histogram in this slide highlights that drug adverse events, or AEs, are a major problem. What it shows cumulatively is that there are over 21 million AEs reported in the USA FDA Adverse Events Reporting System, or FAERS. And this is just the tip of the iceberg, because most AEs are not reported to any agency. It turns out that acute pancreatitis is a serious drug AE. And drugs, as Maisam showed earlier, are a leading risk factor for acute pancreatitis. And these are attested from both our data back at Yale New Haven Hospital from now like 13 years ago, and data more recently shown on the right from Maisam. And note, in both scenarios, drugs were not a mutually exclusive risk factor, but could contribute to additive risk factors. And one reason that drugs are a leading risk factor in children is that children are more likely to take drugs that are associated with pancreatitis, as shown from our own study on the left. And the common pediatric drugs are valproic acid for pediatric epilepsy and asparaginase for acute leukemia, which is much more common in children. On the right is a list from an extensive literature search performed by the Hungarian Pancreatic Study Group, which corroborates that these two drugs are at the top of the list of drug-induced cases. I'd like to focus your attention in this story on asparaginase-associated pancreatitis, or AAP, as a prototypic example. And it's really great to see that there are members in the audience who have also taken on this cause. And the reason is that asparaginase is a crucial chemotherapeutic for the most common cancer in children, which is acute lymphoblastic leukemia. The drug was instrumental in transforming survival from 10% to now greater than 90%. The drug depletes circulating asparagine, which leukemic cells are unable to synthesize de novo. However, pancreatitis develops in 5 to 10% of users, and 1/3 of cases develop severe disease, which is likely the reason why Maisam found more severe cases with drug-induced pancreatitis. Well, a systematic review that we and others performed recently revealed only limited risk factors for AAP, which are shown here in this slide. There was no utility of pancreatic imaging, serum enzyme levels, or genetic screening to predict AAP. Thus, the existing clinical data failed to predict the problem. I'd like to highlight experimental work performed by Julia and Oleg Gerasimenko and Oli Peterson, who are in the audience, and colleagues, where they generated a model of acute acinar toxicity, albeit with fairly high concentrations or dosing of asparaginase, ex vivo in acinar cells. They showed that it evoked pathologic intraacinar calcium signals, and in an in vivo model that was described over many parts of the Q&A. Well, notwithstanding some of the experimental work, what we recognize is that the existing clinical data about AAP fail to predict AAP or to explain its underlying mechanisms. And there was a major unmet need to decipher the mechanisms of AAP and to come up with methods to predict and rescue or prevent AAP. So we decided to take a systems approach, which was unbiased. And it's like putting together pieces of a jigsaw puzzle. First, I'll tell you about clues from the FDA and electronic health records. Second, we'll look at records from results from a case control study of metabolomics along with dietary patterns. Third, I'll show you changes with asparaginase exposure in mice. And I'll try to do this all within the time. We started with the FAIRS for clues using a novel tool called AIRS Mine, developed by collaborators at Cincinnati, shown below. The premise is that drugs can be assigned a risk score for an adverse event. And we can exploit the fact that concomitant drugs reported to the FAIRS can potentially lower or increase the risk score of the adverse event. You can see in the heat map on the left of pancreatitis risk with drugs that reduce the heat map, or in other words, reduce the risk of pancreatitis. And while some of the immunosuppressives show up as promising candidates, the green arrow points to a surprising entity, which is vitamin A. Now, if we home in on vitamin A, the heat map on the right shows that vitamin A exposure appears to mitigate AAP. And the leftmost column is asparaginase alone, which has a fairly high risk of pancreatitis, which is expected. But what is interesting, shown in the middle column, is that independent of asparaginase, vitamin A exposure alone seems to reduce the risk of pancreatitis reporting to below 0. And on the right column, you can see that the few reported cases of a combination of asparaginase and vitamin A have a risk score of 0. And this was our first clue to vitamin A. We next turned to a large electronic health record called TriNet-X. And here we identified two groups, one that took asparaginase and in whom vitamin A exposure was excluded, and a second group that took asparaginase and had vitamin A exposure. Acute pancreatitis was identified by physician diagnosis coding. What we see firstly, as shown by the black bar, is that the frequency of pancreatitis among asparaginase users without vitamin A is close to 8%, which is within the frequency range in the literature. And secondly, shown in the maroon bar is that vitamin A exposure mitigates AAP with an odds ratio of 2. Vitamin A refers to a group of fat-soluble retinoid metabolites which modulate several organ system functions and fundamental cell biological processes, as shown here. And they occur most commonly through ligand binding of retinoid-related receptors that affect transcriptional gene regulation. And vitamin A is derived in the human diet from both animal and plant sources. To probe the role of vitamin A in AAP, we asked whether there are changes in the retinoid metabolites or related pathways that differentiate patients who go on to develop AAP from the controls. And for this, we devised a case control design and performed an untargeted metabolomic analysis. Here is a schematic of the case control design with samples and metadata derived from the Dana-Farber ALL consortium led by Louis Silverman, shown here. The cases went to develop pancreatitis in most situations 3 months after induction therapy, that is 3 months after the first exposure, and repeated exposures of asparaginase, as shown in the histogram below. This is not an acute problem. This happens over time. Plasma samples were taken at 2 time points, firstly at baseline prior to induction, and secondly, right after induction therapy. After observing that there were no differences in the metabolites between the 2 groups at baseline, we next asked whether there were differences in the metabolites between the cases and controls after just 1 exposure to asparaginase, which importantly is still prior to the onset of pancreatitis in most cases. What we discovered was striking, which was that the plasma retinoid precursors were reduced in abundance in the AAP cases. And this is shown in the volcano plot. The red arrow points to the pro-vitamin A precursors, the carotene diols. Furthermore, as you can see in the enrichment plot, and as denoted by the red arrow, conveniently, pathway analysis revealed vitamin A metabolism as a top hit. We were fortunate to have from over a third of the cohort metadata on dietary intake at baseline from the start of chemotherapy with this food frequency questionnaire that was done for 30 days, and then during the induction period of 30 days. So we were able to ask whether the AAP cases have reduced dietary intake of vitamin A compared with the controls, either at baseline or during induction therapy. And these data were provided by Elena Ladas from Columbia University. Firstly, we found that there were no significant differences in vitamin A intake at baseline between the cases and controls. And this is a very rigorous food frequency questionnaire that goes through multiple components of the vitamin A micronutrient foods. However, we were intrigued to find that the AAP cases had reduced dietary intake of vitamin A during the induction period, specifically, as shown in the violin plots here. What is particularly interesting and could be relevant is shown on the rightmost graph, that the AAP cases were taking a borderline recommended daily amount of retinol activity equivalents of vitamin A. And you can see the difference. And even though on the plot it doesn't seem like a whole lot, this is a fairly substantial change for the recommended daily allowance. We next asked whether asparaginase as a single agent leads to a reduction in retinoids. And for this, we turned to animal studies. And these are studies from collaborators at Rutgers, shown here, Tracy Anthony and Loredina Quattro. They found that asparaginase, given over a week period as a single agent administered to mice, causes a reduction in serum retinol. And the trend of reduction occurs independent of age, sex, or diet. Here is a summary of the data. Firstly, from the FDA and EHR, that AAP risk is reduced with concomitant exposure to vitamin A. Secondly, from the case control studies, that the plasma retinoids are reduced in the cases compared to the controls after induction therapy. And importantly, that the cases had reduced dietary intake of vitamin A during induction. Thirdly, that injecting mice with asparaginase reduces circulating retinol, and overall retinols reduce the risk of AAP. Here's our working hypothesis, which is still work in progress, for the mechanism underlying the emergence of AAP. Circulating vitamin A is necessary for maintaining pancreatic health. And this is a first finding. Patients with ALL who take asparaginase have reduced availability of circulating vitamin A to the pancreas. And we don't know why. We have some clues, but we think the circuit's broken. However, patients develop AAP only when they have an additional hit, when their dietary vitamin A intake during induction is reduced, as denoted by the red arrow going down. And that probably accounts for the reduced carotene diols. Thus, we propose a multi-hit hypothesis that the combination of asparaginase along with reduced dietary intake of vitamin A predisposes ALL patients to AAP. And the exciting implication of this, as denoted by the green upward arrow, is that dietary vitamin A could prevent or rescue AAP. By using AAP, and this is my last slide, as a prototypic example, we can envision a future for a personalized approach to the global problem of drug adverse events in which the clinician inputs history and anthropometric data, genomics, metabolomics, potentially other targeted omics in the electronic health record, and what is automatically outputted for that particular patient based on the data are what drugs to avoid or to use judiciously, or what prophylaxis to employ in order to avoid the complications of the drug adverse event. So I thank you for your attention.
Can I ask a question? Again, also on Zoom. Vitamin A is a very profound modulator of the immune system, especially Th9, IL9. Any idea what all the treatment, asparaginase and vitamin A, have on the immune system in these patients?
Yeah, thanks, Shmuel, for that question. That is something we're very interested in because of the fact that this is a sterile inflammatory condition. Maybe this is just an example for a broader application in other sterile inflammatory conditions. What we do know, so I don't know the answer, but what we do know is that with a sort of open data set, open source data set, we are able to tease out chemical perturbins that seem to downregulate the gene upregulation that's seen with pancreatitis as well as with asparaginase. And some of them target very specific retinoid receptors. And we think that they might be involved in the immune response.
Thanks.
Yeah, very nice talk. Very nice talk.
Go ahead.
Okay, I'll
hear you. Sure.
Okay, thanks. It's just a comment really in this our work on asparaginase-induced active pancreatitis. We used in vivo and in vitro and show that carbohydrate galactose actually reduced dramatically all hallmarks of acute pancreatitis, particularly efficient for inflammation and necrosis. Necrosis taking them to nearly control level. And this is a freely available drug, and very efficient, just the oral usage is very helpful for pancreatitis.
Yeah, well, thanks. You know, I've fascinating work. I read it with great intent, and I think that there may be multiple protective mechanisms in the pancreas, and obviously one of them is bioenergetics, but another is this whole area of gene expression that somehow provides a protective angle. And I think we stumbled upon that pathway. It may be related, it may be independent. There is, by the way, calcium signaling from retinoic acid, including on CD38, which is a cyclic ADP ribose generator. So, I mean, it's possible that there are intersections, but we think, at least at the moment, these are parallel protective mechanisms.
So here, it's because I don't know if you see me. Anyway, you hear me, right?
I can hear you.
Okay, so it's a very, very, very well presented talk. I really enjoyed this. So this is either one of the most exciting findings in recent pancreatitis history, or this is a complete dead end. Because, I mean, you agree that data so far is very circumstantial, right? And then what's really needed is animal model experiments to nail this, really.
Well, I mean, I might not put it that way. Nicholas, you certainly have a way with words. I would say that a systems approach is where you take multiple points of evidence and try to put it together. But you are right. The next immediate steps are to examine whether vitamin A supplementation can improve pancreatitis in various models, or whether vitamin A deficiency or deficient states predispose, and with asparaginase. I think one of the most immediate steps, which we're trying to convince the oncology groups to do, is to undergo something similar to the effort, which is look at a healthy diet that has at least above the RDA for RAEs, retinol activity equivalents, compared to the junk food that kids eat during induction therapy.